Manoa Reservoir in Box Elder County, Utah, is located just east of Brigham City. The reservoir is a beautiful and popular spot for recreators, for fishing, boating, and even swimming. The reservoir is not, however, always this pretty. It is threatened yearly by excess nutrients causing harmful algal blooms. Harmful algal blooms occur when cyanobacteria, also known as blue-green algae, grow in water. They are considered harmful due to their potential to emit cyanotoxins, which are harmful to humans and animals. HABs frequently occur in Manoa Reservoir in the late summer and early fall and have caused the health department to issue warning and danger advisories and even closures. The remediation of these algal blooms is the goal of Envision Environmental Solutions.
Hi, I'm Carolina Williams, and I'm from Oklahoma.
Hi, my name is Tyler Wallace, and I am from Declo, Idaho.
Hi, my name is Stephen White, and I'm from South Jordan, Utah.
Hi, my name is Jeffrey Kennedy, and I'm from Boise, Idaho.
We chose to look at the issues facing Manoa Reservoir because of its closeness to Cache Valley. It is a popular spot visited by some among our team. We recognize the importance of the lake for both the cities of Manoa, where the lake is located, and Brigham City uses the lake for irrigation water. Our project consisted of determining the problems facing the reservoir. We took samples throughout the summer of 2020 to later analyze for nutrients and cyanotoxins. The reservoir is bordered to the south and to the west by the community of Manoa, the bulk of which you see here. This is a small town whose major land use consists of agriculture, with croplands and pasture. The land cover present in the watershed provides an idea of what nutrients may be released into the waterways and where they may be coming from. An analysis of the land cover provides an estimate for the distribution of nutrient sources. Significant agriculture is present in the watershed, which typically releases much more nutrients than undeveloped forest land or developed residential land. This agriculture provides roughly half of the estimated nutrient loading from land cover. Other nutrient loadings can be identified at specific point sources. The only permitted source in the watershed is the Manoa Fish Hatchery. Since 2000, the hatchery has put into effect practices to reduce its nutrient releases, such as building a settling pond and reducing phosphorus in the fish feed. The hatchery is located south of the reservoir and releases its effluent into Maple Creek, which flows into Site 4 on this map. The inlet of this creek became a sampling point due to the hatchery. Other points were chosen by identified inlets or points of interest. Site 7, for example, is located at the boat dock on the lake. Site 0 was added during the sampling process as water began to flow into the lake. Other sites were identified as points of interest early on, but were ultimately not used in sampling collection. Sampling of these sites allowed us to quantify nutrient levels at different points of the reservoir, as well as map changes over time. Feel free to ask us about our findings. We learned a lot from this process, both about the lake, as well as sampling procedures in general, and we might have some good stories to share with you too. A major part of our data analysis relied on GIS software. We identified the drainage area for the reservoir, as well as the rivers and canals present in the watershed, seen here on the left. We determined that a very large portion of the watershed drains into only two specific locations on the reservoir, both of which are included as sample sites. We have Dam Creek to the north, which drains into sample site 1, and Maple Creek from the south includes a drain from the fish hatchery into sample site 4. After identifying the predicted sources of nutrients in the watershed, we proposed a solution to preventing HABs from occurring in the reservoir. Watershed management is required to reduce nutrient loadings into the reservoir, but several solutions exist for treating and removing phosphorus, as well as targeting and preventing the growth of the specific cyanobacteria involved. We will discuss our proposed solution of ultrasonic treatment during Senior Design Night on May 4th. Come learn more as we present our full project.